Our final section of chapter 12 talks about how does the DNA molecule actually replicate itself. Before we go into replication, let's just speak for a moment why organisms, why living things look so different since they all have the same four base pairs. And it all has to do with the arrangement. And as we'll see as we look at the DNA makeup of different organisms, the differences between us are very small, but our outward appearances are very, very different. So it's the sequence of the nucleotides that determines who's who. And just a small change in those sequences can cause drastic results. So let's go back, way back to the beginning of biology and look at a prokaryotic cell. And if you remember, a prokaryotic cell does not have a nucleus. So where is its DNA? Its DNA just floats freely in the cytoplasm, and generally there's only one strand of DNA. But you can see this is an E. coli, a type of bacteria that lives in our gut. And this particular bacteria, you can see its genetic code on the right, and that the base pairing occurs just as we would expect it to. Eukaryotes, it's a little bit of a different story. A eukaryotic cell is much more complex than a prokaryotic cell. It has a nucleus, it has many organelles that you're not going to find in those single-celled organisms, and we're looking at 1,000 times the amount of DNA. And that DNA is going to be housed in the nucleus. And it's interesting, if you look at this chart below that just shows a few species, you would think that human beings, we consider ourselves, the top of the food chain here, to be the most complex. But you can see that that is not true if we look at the number of chromosomes. A duck has almost twice as many chromosomes. A rat, pretty darn close, and a tobacco plant has more chromosomes in it than an actual human. So it's kind of interesting. All right, let's take a look at a couple new terms that we're going to use to describe this piece of DNA and all of the little parts that go into making it a chromosome. So if we start down here, we can see our base pairs and our sugar phosphate ladder that's twisted in the double helix that we call DNA. Well, that DNA is actually wrapped around protein molecules called histones, and as those get wrapped up, they make these coils that are eventually going to become chromatin, that will become a chromosome that lives inside the nucleus. So let's take a closer look. So we know the chromosome itself contains DNA and protein. The spaghetti-like structure that makes the chromosomes is the chromatin. The chromatin is actually made out of DNA wrapped around histones, which are just protein molecules. Those are called histones. And when you have this DNA wrapped around histones, we call it a nucleosome. So down here we have just our DNA. It gets wrapped around a histone, becomes a nucleosome, these thick coils, which condenses into chromatin, which condenses even more into a chromosome, which will end up inside the nucleus. And this is simply another look at it, just a slightly larger picture. We can start, I guess, from this side where we've got a chromosome. We can look at the chromatin. We've got our coils going on here. If we look deeper into the coils, we can see that those are nucleosomes, which are just pieces of DNA, though the strands of DNA wrapped around the histones, the proteins, and then if we go even smaller, we get our double helix structure. So when a cell is preparing to divide, it has to make an exact copy of its DNA. And it's important that this exact copy gets made, or the offspring are going to be genetically different from their parents, and we certainly don't want that. And the process itself is called replication. Replication, prokaryotes, and eukaryotes, always occurs during interphase, and it occurs during that S phase of interphase where we're getting ready, everything ready to go into cell division. And in prokaryotes, where we only have this one single strand of DNA, there is a particular spot, and it's a special point where replication will begin. And it will not start until a regulatory protein comes in and hooks into this spot, and then replication begins in both directions. In eukaryotes, it's slightly different. Remember, we have upwards to 1,000 times the amount of DNA, so it would take much, much too long if we had just one spot where replication occur. So there's hundreds of places along that DNA molecule where replication can take place. And it will start at that particular spot called a replication fork, and DNA goes in both directions. And it'll continue until the entire strand is covered. And each of these little places where the replication is occurring, the DNA molecule has to separate, come apart. Those are called the replication forks, and we're going to take a closer look at those. So you can see in this picture that we've got a replication fork at this location and this location, and that in between both of these we have replication going on. So we have base pairing taking place, and new strands of DNA being paired up with their old strands. So how does this all start? Well, it all begins with an enzyme. So we have our parent DNA molecule, and it has to unwind, and it has to come apart. And there is a special enzyme called DNA helicase that comes in, and remember, enzymes you can tell with that ASE ending, and it acts like a zipper. And it's going to travel down the molecule in this direction, unzipping the molecule and splitting it apart. So this picture is going in the opposite direction. You can see our molecule is getting unzipped this way. And inside the nucleus, we have our bases, just kind of hanging out in the nucleus, waiting to be called to form our complementary molecules. So these nucleotides are going to be formed by these bases that are just kind of floating around in the nucleus. There is a second enzyme, and this enzyme is very, very important, called DNA polymerase. And what this DNA polymerase does is it comes along and it takes these bases, these nucleotides, and it attaches them in. So this is very exaggerated, but the DNA polymerase kind of grabs both sides of the molecule, slides along, and you can see that we're putting, hooking up in this one here, we've got our blue guanine matching up with the green cytosine. You can see that thymine is next, and adenine is getting ready to pair with it. And what this DNA polymerase is doing is it's hooking them together, and at the same time it's checking to make sure that the base pairing is matching up correctly. It's to work pretty hard because about 80 nucleotides are added every second in your body. So it's kind of amazing if you stop and think about it. So when we're done, here's just another look at it. We've got our helicase that's unwinding. We've got our replication fork at the point where they are hooking in. We've got our, you can see our complementary ends, our 3' and our 5' ends. And we've got our DNA polymerase moving along and checking it. And then this picture gets a little bit more complicated, more than we care to do right now. But basically, what you need to take away from this is that when we are done, we have 2 exact copies of the DNA, and each one of those copies has an old piece from what we would call the parent, and then a new piece from what we had just made during replication.